Chef Cochell, we got the cocktail cantina. I'm just wondering if other people want self-efficacy. I just realized it's not really a threat at all to be yourself, plasticity, and the CNS, learning from experience. The third principle of this chapter states that the CNS responds to experience by learning. That is, it reorganizes some of its neurochemistry and connections so that the experience is remembered. It's very important to understand that this plasticity is a broad concept. Not only does the CNS remember events that are consciously experienced, but it also changes in response to all sorts of signals such as constant presence of drugs. The most familiar plasticity in the CNS the simple remembering of experiences, faces, others, names, classrooms, lectures, and more. I remember this jokes. Neurological mechanisms, which this kind of learning happens or not completely understood. We have some clues. One important side of learning appears to be miscoordinated love. It's just those synapses of nerve cells are quite complex, and there's extensive biochemical machinery in both the presynaptic and the postsynaptic areas. We think memory is built on one synapse at a time. Some synapses are stimulated repeatedly, change how they function, learn and maintain the change for a long time. There's an electrical manifestation of this learning that scientists call long-term potentiation, as a long-lasting strengthening of the electrical signal between two neurons that occurs when the synapse between them is stimulated. We're not sure how this happens, but it's likely through a series of the chemical changes and how the first neuron releases its neurotransmitter and how the second neuron responds to the neurotransmitter. Depotentiation can be produced by prolonged slow activity or by a very strong high-frequency activity, like that which occurs in seizures. It may be a protective mechanism by which the CNS prevents a seizure or brain trauma from encoding new information into the circuits. Again, it is almost certainly done under the control of cell signaling pathways, and thus could be manipulated by drugs. This gradual change in the electrical strength of the connection seems subtle, but it makes intuitive sense that the memories could form in this way. Can the brain actually change physically? We used to think that once a person was mature, the brain didn't change anymore. However, more and more research shows that actual changes in the shapes of synapses also can happen in response to earlier experiences. We know that the shape of certain neurons in the brain change when different hormones become available. For example, at least in animals, treating them with hormones can stimulate the production of little protuberances or spines on the dendrites of neurons. Other research has shown that the synapses actually remodel themselves over time after different levels of activity, so actually get lost or remade. For example, prolonged stress seems to actually shrink the dendrite of neurons. Perhaps the cognitive difficulties people encounter during prolonged stressful periods. It has been known for a long time that this happened in lower animals. For example, as songbirds learn new songs, the structure of certain parts of their brain change. It was once thought that the brains of animals did not have this type of structural plasticity. However, more recent studies have shown similar changes in rats. And scientists think that they probably occur in all animals. The most exciting reason in neuroplasticity is the understanding that the brain can actually make new neurons. This process of neurogenesis was thought to occur mostly during prenatal development. Now, what is happening in adult? I usually have one lined up. As normal ones happen in lower animals, for example, as the songbirds alludes to song. There have been some intriguing findings regarding the effects of drugs on neurogenesis in animals. It appears that depression reduces neurogenesis and subsequent treatment with antidepressants restores it. More relevant to this book, the laboratory of Fulton Cruz at the University of North Carolina has made the startlingly discovery that binge exposure to alcohol dramatically increases rat production, particularly in the forebrain, which is a rapid brain area in rapid development. The practical implication of this are enormous because adolescents tend to be binge drinkers. and this behavior impairing their brain development. What other drugs affect these processes? Does this happen in humans? These questions should and will be answered by future research, but they now alert us as a possibility that drug abuse have profound effect on teenage development. Do all parts of the brains learn? The process we describe do not happen in just one part of the brain. There are indeed specialized neural networks, especially in a part of the brain called the hippocampus, where learning occurs and memories are created. People who have bad damage to this area of the brain cannot learn new things, although they can remember things that happened before. However, most forms of plasticity can occur all over the brain and affect all brain function. For us to be able to function normally, all brain processes need to proceed unimpaired. All the neurotransmitter system need to be working. The brain needs to change with time to reflect previous experience, that is, to learn to restore balance if it is over or understimulated. The developing brain. While the brain of adults change all the time, what goes on in adults is trivial compared to the phenomenal change that occurred while the brain is developing. The brain assembles lists careful throughout the process, neurons growing out, and through chemical signals around them, gradually finding their way to the correct destination, where they make the connection that they need to maintain. During this time of life, for example, a drug called APP5 blocks a certain subtype of the exciter neurotransmitter glutamate. This particular subtype, the MDMA, has a very special property letting calcium into the cell only when the cell is receiving excitatory signals through the other synapses. The calcium causes LTP to occur at those synapses, thus the MDMA receptor is likely a memory switch. When the cell is receiving a signal and the MDMA receptor is activated, the cell remembers the signal by processing, strengthening that synapse. It is wonderful that we've been lucky enough to find the MDMA receptor because it appears to be one of the most important receptors for learning and other forms of neuroplasticity. It may teach us about how memory occurs and how some drugs disrupt it. For example, in laboratory experiences, if we block the MDMA receptor so that glutamate cannot bind there, LDA doesn't occur. Rats do not learn mazes. This is every reason to believe that learning and neuroplasticity are also suppressed in humans. Alcohol in rats block MDA receptors, suppress LTP, and suppress maze learning. So now we know why we forget and when we get drunk. Many of the effects of the ability to learn, there is no question about it. But which drugs have which effects and for how long? One of the best stories about the effect of drugs on learning was told me by a drug company representative during an airplane trip. It seems that some of the professional staff from this company were making a quick trip overseas to a meeting and they needed to sleep during the plane ride because their lecture was scheduled as soon as they were to arrive. So this group had a few alcoholic drinks and took over their newly marketed sedatives, benzodiazepine, get to sleep. Everything went well, including the lecture, and the scientists returned home in a couple of days. The only problem was when they returned, they remembered nothing of the meeting, not their lectures, nor of anyone else. By far the most common. Now we know that LTD and depenetration is easy to imagine there would be reasons for the CNS to reduce activity in some particular ways and thus forget some neuroplastic changes. Therefore, it is completely reasonable that some drugs could enhance this type of signaling and reduce their ability to learn. On a broader note, neurologists are exploring ways to use drug therapy to enhance learning. This research is particularly important for the many people who suffer from Alzheimer's disease or other brain disorders that impair learning. Most of the rest of us have also realized the ability to learn faster. There are many tantalizing clues that this may be possible. One of the most interesting clues comes from an experience that almost all of us have had. It's the do you remember when you were doing when question. Every generation has at least one of these questions. For older people, is what they are doing when they heard that JFK was assassinated. Nearly everyone recalls the fateful morning of 9/11. Think of an example, the first time you had a very important and emotional experience, either positive or negative. Why is it that we remember some experiences so well, and not only the event, but what clothes we wore, what the room looked like, and what we ate? Ongoing experience shed a lot of light on this phenomenon. Dr. James McGough at the University of California took two similar groups of people and placed them in separate but similar rooms with all sorts of cues or decorations. I wrote a song about 9/11, dude, and donated blood. I wrote a song about 9/11 and donated blood. The goal is the subject that groups do an emotional story and see how they remember the story and the environment in which they experience the event. I don't know what it was called, dude. What this experience introduced was given a drug that blocks a particular subtype of adrenaline receptor.